Mariana Kaplan. She's an associate professor in the Division of Rheumatology at the University of Michigan. As a kind of introduction of her research area, her work has till now focused on identifying mechanisms that promote end organ damage in lupus, particularly premature cardiovascular diseases. More recently, her research has focused on the role that aberrant neutrophils and aberrant neutrophil extracellular traps play in the pathogenesis of various autoimmune diseases, including lupus. In this area is enclosed the speech today, which is entitled Net Relevance to Lupus Pathogenesis. Please, Dr. Kaplan.
Thank you very much. I want to start by thanking the organizers for the kind invitation. And I will try to give an overview of what we believe could be the role of NETs in lupus pathogenesis. So the way we view lupus nowadays is as an autoimmune syndrome where a variety of factors, including genetic and environmental conditions, may modulate the onset, the severity, and the clinical phenotype. And we also understand better now how abnormal innate and adaptive immune responses may play key roles in initiating and perpetuating this syndrome. A sine qua non in lupus is the formation of autoantibodies and immune complexes, primarily directed to intracellular nuclear antigens. And we also know that all these abnormalities lead to a clinical diverse phenotype of acute and chronic manifestations. And as our patients live longer, we are now seeing more and more the effect of chronic inflammation and chronic oxidative damage in the development of end-stage complications, including renal failure and atherosclerosis. And a key question for many years has been how do these self-intracellular antigens become immunogens that can trigger and maintain strong and prolonged autoantibody responses? And for a good number of years, research has primarily focused on the role of aberrant apoptosis and impaired clearance of apoptotic material as a key phenomenon that externalizes and modifies these autoantigens. And I think this diagram continues to be true, where, again, aberrant apoptosis, impaired clearance plays a prominent role in externalization of autoantigenic material that leads to the formation of immune complexes and autoantibodies, induction of type 1 interferon responses, and induction of abnormalities in adaptive immunity. But perhaps this model has been somewhat simplified or oversimplified, in part because we didn't know much about what other types of cell death, we didn't know about the existence and the role that other types of cell death may have in triggering of autoimmune responses. And this has become much more complex as we are beginning to understand how other types of cell death may have anti-inflammatory and pro-inflammatory effects, how some types of cell death may be specific for some cell types, as we believe is the case of, for example, pyroptosis, a caspase-1 dependent process where that is primarily observed in dendritic cells and macrophages. And, of course, considering that all these processes, if aberrant, may be involved in breaking tolerance and inducing autoimmune responses in predisposed individuals. And, of course, the role that enhanced cell death or aberrant cell death may play may be also related to what cells are dying or in which way they're dying. And if we think about human neutrophils being the most abundant white blood cells and having a very short lifetime, and thinking that 55% of bone marrow weight is devoted to neutrophil production, we can envision that abnormalities in cell death in a cell that also has a very abundant pro-inflammatory machinery inside the cell could potentially play potent deleterious roles. So this novel form of cell death was identified in neutrophils that I will discuss today. Was identified first in 2004 by Arturo Sichlinski at the Max Planck Institute in Berlin. And it was described as a part of the antimicrobial strategy of the neutrophil, where you can see that there early on phagocytosis occurs shortly after exposure to microbes. The granulation may occur about 30 minutes after exposure to microbes. And then there was a description of this more protracted event where there is a mix of the nuclear material and the antimicrobial proteins present in the neutrophil granules that are then extruded during a process of cell death that was described as netosis. And these the nets that you see here in a very nice picture when they were originally described are then indeed a mixture of histones and chromatin and nuclear components with the granular components, the potent antimicrobial proteins that are present in the granules. So this was described initially as a potent machinery that the neutrophil had to trap bacteria and potentially to also kill them. And this has become much and more complex, and we, I think, are only beginning to understand how nets are formed, what are the different ways by which the neutrophil extrudes the nets, and what are the molecular pathways involved in this phenomenon. So as I mentioned, the original description by Sichlinski's group was that this represents a novel form of cell death that is distinct from apoptosis and necrosis, and where there is degradation of the nuclear envelope and citrullination of histones, which allow the chromatin to decondense. And after there is the degradation of the nuclear envelope, there is a mix of this nuclear material with the granular proteins, and then rupture of the plasma membrane with lysis and net release. And this is highly enriched in histones and proteases that can be potentially toxic to other cells. But then there have been other groups, and there is still a lot of controversy about whether all netting cells die or whether other processes occur before the cell die. But for example, Paul Kube's group has described this process where the neutrophils become enucleated during the process of net formation, and the cell still survives in the absence of this nucleus, sort of like what you will see with other enucleated cells like erythrocytes. But you can still see chromatin in these cells, and then eventually this leads to net release. And furthermore, there is also some evidence from a couple of groups suggesting that mitochondrial DNA may also be extruded during net formation, and this may allow the neutrophil to survive at least for a while before overt cell death occurs. This has become also more complex because initially most of the manuscripts focus on microbes triggering nets, and we know now that there are a variety of microbes that can lead to net formation. But we have also been learning more about unquote, sterile conditions that may trigger netosis. For example, platelets under condition of flux may be potent inducers of net formation, and certainly there are a variety of cytokines that have been recently reported to be strong inducers of net formation, including IL-8, TNF-alpha, potential interferon alpha in the presence of complement components, etc. The initial description mentioned that in order for netosis to occur, it was required that there was a functional NADPH oxidase activity, and that this was a key phenomenon in the formation of nets. And this may continue to be true, but I think there is more evidence coming from various groups that not all nets may be created the same way, and that there are conditions in which NADPH oxidase activity is not required. The other thing we know about the pathways of net formation is that we need the presence of this enzyme, peptidyl arginine deaminase 4, which is one of the paths that is present in the nucleus of neutrophils and mediates, as I mentioned, citrullination of histones, which is considered key for the formation of nets. And I'm mentioning this because it will become relevant when we talk about lupus. And finally, and I think before I start talking more directly about lupus, I think this is important when we talk about vascular damage in autoimmune diseases. There is recent literature from several groups suggesting that NETs may be important players with regards to endothelial cell damage and thrombus formation. So NETs carrying a variety of toxic molecules, particularly histones, but possibly other granular molecules, have been found to be strong inducers of endothelial cell death. And also there have been a variety of, a number of papers primarily focused on venous thrombosis that have described that NETs become an important area for platelet activation and also for coagulation factor activation, and that neutrophils are required in these models of this venous thrombosis for clots to occur. So I would like now to spend some minutes discussing some of the abnormalities that have recently described with regards to neutrophil phenotype and function and the potential role that NETs could play in inducing autoimmune responses and organ damage. So this is data from Virginia Pasquale's group now 10 years ago, where studying pediatric lupus patients, she identified a strong granulocyte signature present in the PBMCs from these patients. And this was, in a way, surprising because neutrophils are typically not present in PBMCs from healthy controls. And this was at the time associated to the presence of an immature granulocyte, that were present in the PBMC fractions. And also in 2008, there was another paper where bone marrow biopsies from lupus patients were analyzed, and it was found that strong granulocyte signatures in the marrow were associated to lupus disease activity. And some years ago, we became interested in studying more in depth these low-density granulocytes that in our hands are present in all samples from adult lupus patients, and at different levels. Some patients have very high levels, others have small percentages of these cells. But these cells, as I mentioned, are present in the PBMC fraction. We name them low-density granulocytes. And we have found that they express, their nuclear morphology is indeed consistent with a more immature form, but they behave like mature neutrophils when we talk about expression of cell surface markers or some of the functions that mature neutrophils should display. And we have been interested in trying to assess whether these low-density granulocytes have pathogenic roles in lupus. So one of the observations we initially made were that these cells appeared to be more pro-inflammatory. They secrete higher levels of a variety of pro-inflammatory cytokines, and they were also able to synthesize increased levels of type 1 interferons upon specific sources of stimulation. Of course, if you look at cell-per-cell basis, the amount of type 1 interferons this cell may make may be much less than what a plasmacytoid dendritic cell would synthesize, but there are many more neutrophils in the circulation and in the marrow than plasmacytoid dendritic cells. So we propose that these cells potentially could represent an important source of type 1 interferons in specific microenvironments in lupus patients. The other thing we have assessed is the gene expression profile of these low-density granulocytes. And what we thought was interesting was that when you did microarrays from these low-density granulocytes from autologous normal density lupus neutrophils and from control neutrophils, we found that if you take normal density neutrophils from lupus patients and compare the gene array expression to those of control neutrophils, we saw absolutely no differences, which was surprising to us. But then there were distinct differences when you took these low-density granulocytes. We did not see evidence that these cells had been exposed to higher levels of type 1 interferons. They do not express an enhanced type 1 interferon signature. But they clearly overexpress various mRNAs that are involved in, that are associated to proteins that we see present in granulocytes, in the granules of neutrophils and in the nets, including the cathelicidin NL37, MPO, cathepsin G, etc. So the most overexpressed signature in these granulocytes is actually of genes, of molecules present in azurophilic or primary granules, which are synthesized at early stages of development, again indicating that this could represent a more immature form of neutrophils. So we then became interested in assessing whether these low-density granulocytes had a different capacity to form nets, and whether these nets may represent a source of autoantigens, but also play roles in end organ damage. And more or less at the same time, there were a number of papers that addressed the issue of nets in lupus and other autoimmune diseases, and this is becoming probably more and more complex and interesting. The first manuscript was also from Sichlinski's group in Germany, and similar to what has been described in apoptosis, a question now becomes: is there an imbalance between net formation and net clearance, and could this be involved in leading to prolonged exposure of modified autoantigens? And at least from what we know nowadays, in contrast to apoptotic cells, it doesn't appear that netting cells lead to eat-me signals that are recognized by other cells and lead to engulfment of this netting material. This may change in the future, but what we know nowadays is nets are primarily degraded by nucleases present in serum and other fluids. And what the group in Germany found was that about a third of the serum from patients with lupus had impaired capacity to degrade nets. And they associated this impairing degradation to the presence of possibly anti-net antibodies or inhibitors. This was described in a general term at the time of what these anti-net antibodies may represent. And they also showed a strong correlation between having impaired net degradation and developing lupus nephritis. With the same story of impaired net clearance, work from Anna Blom's group in Sweden found that nets actually were strong activators of complement, and that C1q depositing in the nets was actually involved in inhibiting net degradation. So potentially identifying nets as an amplifier of disease by activating complement components, complement pathway. When we talk about enhanced net formation and what this may do to regards to SLE pathogenesis, the very elegant work by Michel Gilliet's group identified that netosis was enhanced in SLE neutrophils, and that the patients with lupus had the formation of DNA LL37 immune complexes, and as I mentioned, LL37 is a molecule that is present in the neutrophil granules and that is externalized during net formation, and that these DNA LL37 complexes actually permitted DNA to be protected from degradation by nucleases by the presence of this LL37. And whether LL37 is the only molecule present in the nest that does this, I think still remains to be determined. But they hypothesized that this was a mechanism that protected DNA from degradation. And they also found that this was a mechanism that led to these complexes of DNA and LL37 were potent stimulators of interferon alpha synthesis by plasmacytoid dendritic cells. Furthermore, they identified that a number of lupus patients had anti-net antibodies or anti-LL37 antibodies and other antibodies directed to antimicrobial peptides, suggesting that the formation of nets could indeed lead to B-cell activation and the formation of antibodies that also had the capacity to further promote net formation. And similar findings were reported by Virginia Pasquale's group, and in her case, in pediatric lupus patients, what her work suggested was that interferon alpha may act as a priming for net formation, but this was triggered when by anti-RNP antibodies, and that this mechanism was involved again in the induction of interferon alpha synthesis by PDCs. What we have found is that when we separate the low-density granulocytes from the normal density lupus neutrophils, the cells that seem to develop a very enhanced susceptibility to form nets ex vivo are these low-density granulocytes. So if we look at normal density granulocytes from lupus patients, as soon as we get them out of the circulation, we really don't see much difference if we don't stimulate them with regards to their capacity to form nets. But you can see that these low-density granulocytes actually come out of the circulation, making a significant amount of nets. And after we cannot further stimulate them to make more nets. If you see what happens with PMA, a strong inducer of nets, both control and lupus neutrophils will make more nets, but these cells are already probably exhausted in their capacity to form more nets because of how significant the enhancement is as soon as we get them out of the circulation. And what we have found is that indeed these nets obtained from these low-density granulocytes have enhanced levels of LL37. They externalize anti-double-stranded DNA as a potential autoantigen. We haven't found evidence that other common lupus autoantigens such as Rho and La are externalizing the nets. Doesn't necessarily mean it's not the case, we just haven't been able to detect them. But they also externalize pro-inflammatory cytokines such as IL-17, and this may be an amplifier of inflammatory responses in various organs, and it's the focus of current investigation. Similarly to what the other two groups have found, we found evidence that these low-density granulocytes when netting, were strong inducers of interferon alpha responses in plasmacytodendritic cells, and that this phenomenon was abrogated when we blocked net formation. And the other finding from these first observations were that we found evidence of netting-like material in organs from patients with lupus. And we particularly focus on the skin with patients that were displaying active skin disease, and also in patients with lupus nephritis. And these netting neutrophils were patients with higher levels of netting neutrophils in the biopsies were actually displaying also enhanced levels of anti-double-stranded DNA, which doesn't prove causality, of course, but it may indicate that this increased netting in tissues could represent a source of, could lead to breaking of tolerance and be involved in the development of pathogenic anti-DNA antibodies. Another key question is, what do nets do to other immune cells? And this is an area that is just beginning to be explored. There is evidence from other groups indicating that nets may actually be priming in adaptive immune cells, maybe playing roles in T-cell activation, and may modulate B-cell responses in the spleen. But I think we'll learn much more as various groups continue to actively work on this. We have recently focused on what netting neutrophils may do to macrophages in the case of lupus patients, particularly interested in what may be happening at the level of the kidney, but also with regards to blood vessels. So something we recently found was that nets are strong activation, and LL37 specifically, appear to be strong activators of the inflammasome machinery. And just as a reminder, the inflammasome is this multi-molecular structure that leads to activation of caspase-1, and caspase-1 activates pro-inflammatory cytokines such as IL-1 and IL-18. And this may be important in inflammatory responses and pathogenic in lupus. And what we have found is that lupus macrophages actually appear to be particularly sensitive to activate their inflammasome machinery in response to nets. But this was also observed in both human and murine systems for control macrophages. And what we have found is that there may be a vicious cycle that among the many ones that may occur in lupus, where the formation of nets may lead to the activation of the inflammasome machinery in the macrophages. But upon release of IL-1 and IL-18, these cytokines may actually further enhance net formation and therefore amplify an inflammatory cascade that may be relevant to end organ damage. And I think we need to explore much more what these nets are doing to other cells, either enhance inflammatory responses or potentially whether there are any immunoregulatory activities of these nets that may be important in down-modulating damage. And and the other key question is that it's also just starting to be explored is whether it's not only the presence of specific proteins in the nets associated to DNA and nuclear components that may be immunogenic, but also whether there are specific post-translational modifications of these proteins in the nets under specific sources of stimulation or in specific disease states that may be potentially pathogenic. We do know that there are a number of net autoantigens identified in various autoimmune diseases, and you can see lupus on the top, as there are quite a few net autoantigens that have been described in lupus. Whether they lead to pathogenic autoimmune responses remains to be determined. So we would have a model potentially where neutrophils and maybe distinct subsets of neutrophils have an enhanced capacity to form net. Maybe the triggers of this enhanced netosis remain to be determined, but certainly autoantibodies and cytokines may appear to be key targets, and whether initial infections in predisposed individuals lead to the initial events of aberrant netosis is also remains to be determined. But this externalization of immunogenic proteins coupled to nuclear material and maybe associated in a subset of patients to impaired clearance may lead to enhanced exposure of modified autoantigens, as well as the induction of direct or indirect tissue damage. Of course, to prove this hypothesis, we need extensive work in vivo, and we need to start with animal models. So I would like to discuss, spend the last minutes talking a little bit about what we know so far about netosis in murine lupus. Some is work published by other groups, and the last minutes I'll talk about some of our work that is not published yet but has been accepted, is in press now, accepted for publication. So P.J. Ott's group in Stanford had a paper in Arthritis Research and Therapy a few years ago where they did analyze the immunogenicity of nets, and as part of the experiments, they took biopsy mice and they immunized them with netting material, chromatin isolated from the nets, and they tried to see whether this could break tolerance. They did not do this in lupus-prone mice, but they took non-autoimmune mice. And what they saw were moderate IgM and IgG responses to DNA and other net antigens, but they saw no evidence of a lupus phenotype. And this, in a way, resembles what George Mevorak and Keith Elkon have found years ago with regards to immunization with apoptotic cells, where it was really in those mice that were predisposed to autoimmunity where they saw a clinical phenotype. So this has not been tested in lupus-prone mice, and the authors commented that these nets that were generated in vitro may not be equivalent to the in vivo generation of nets, and they may be lacking critical antimicrobial peptides that, for example, could activate TLRs and engage in the development of autoimmune responses. I mentioned to you that at least the initial studies with nets suggested that NADPH oxidase activity is required for net formation. So Mark Schlumsky's group last year published a study in which they took the lupus-prone model, the MRL-LPR mice, and they knocked out NOX2 activity, the enzyme. And they published that, conversely to what contrary to would have been expected, these mice actually, you would expect that if netosis was inhibited by knocking out NOX2, that lupus activity would be abrogated. And in fact, they found the opposite, that these mice actually developed more autoantibodies, and they had more severe lupus nephritis. And this could mean a variety of things. It could mean that nets are not required for pathogenesis in this specific model. They did not study other models. It could also mean that, NOx has important immunoregulatory effects. But I also want to remind the audience, this is, in a way, the mouse model of chronic granulomatous disease. These patients are immunodeficient, but they also are pro-inflammatory. They have an autoinflammatory condition that is also associated to immunodeficiency. And I think that's why studying these mouse models can be problematic, because we have a combination of inflammation associated to immunodeficiency. So in a way, it's not and that's hence the name. They develop granulomas, and NOX2 is required for cytokine processing, for example, IL-1. So I think this cannot tell us all the story because of the specific model used, and also because NADPH oxidase may not be required for all forms of nets, and because NOX2 may have important immunoregulatory effects. I would like to show you some of the data that we now have in press studying another lupus model, which is the NZM2328, that has a clinical phenotype similar to the New Zealand black, New Zealand white mouse. And the first thing we wanted to establish in this mouse, which is interferon alpha dependent or type 1 interferon dependent, is whether there was evidence of enhanced net formation in this model, and whether there was presence of anti-NET or anti-LL37 equivalent antibodies in these mice, and then tried to explore whether these NETs were present in organs and whether we could actually try to inhibit NET formation in vivo and see if we abrogated disease activity or vascular complications, which we have studied in depth in this model. So the first thing I want to show you is that it does appear that spontaneously, and we studied here bone marrow neutrophils. Bone marrow neutrophils from the lupus-prone mouse are more prone to form NETs in the absence of added stimulation. And if we stimulate them, they form more NETs also than BALB/C, non-autoimmune prone, non-lupus prone mouse. Then we took serum from, what we evaluated was whether this was present only when the mice were already sick or whether enhanced NET formation could be seen in pre-disease mice. And this is indeed the case. Even when we look at 7 to 8 week old mice, we can see that they have enhanced NET formation. And this is comparable to what we would see in much older mice that have already developed nephritis. And when we took BALB/C neutrophils, control neutrophils, and we exposed them to either BALB/C serum or serum from these lupus-prone mice, we found that the serum from the lupus-prone mice enhanced NET formation in control neutrophils. And indeed, it appeared that this serum has a capacity to bind to NETs formed in vitro. Then we assessed whether these lupus-prone mice had evidence of anti-NET antibodies and antibodies to the mouse ortholog of LL37, which is CRAMP. And what we saw, again, was that, especially in older mice, there were enhanced levels or enhanced prevalence of anti-NET and anti-CRAMP antibodies. And that indeed the serum from the lupus-prone mice, but not the BALB/C serum, appears to bind to a number of proteins present in the NETs. The other finding was that, similar to what we had seen in humans with lupus, we observed evidence of netting-like material in the kidneys from these NCM2328 mice. And finally, and before we tested an inhibitor of NETs in vivo, we took endothelial cells from BALB/C mice and exposed them to isolated NETs or vehicle, and studied the capacity of these endothelial cells to relax in response to acetylcholine. And this is an assessment of how healthy the endothelium is. And the more relaxation, we believe that the endothelium, there's enhanced endothelium-dependent vasorelaxation. Lupus-prone mice, we know, have impaired endothelium-dependent vasorelaxation, similar to the endothelial dysfunction described in humans with lupus. And again, what we found is that, confirming what other groups have described in vitro studies in endothelial cells, is that upon exposure to these NETs, mouse endothelial cells and mouse aorta show enhanced endothelial dysfunction. So I mentioned to you at the beginning of the talk that PAD4 may be important in NET formation because it's involved in citrullination of histones leading to chromatin decondensation during NET formation. And in fact, a few years ago, Yanming Wang and others published reports that PAD4 formation was essential for the formation of NETs upon exposure to bacteria. And they generated these PAD4 knockouts and showed that these mice could not generate NETs. So with this idea, we partnered with Paul Thompson at Scripps, and he has generated a variety of PAD inhibitors. Some are pan-PAD inhibitors, others are more PAD4 specific. And we decided to test whether one of these compounds called chloramidine, which is a PAD inhibitor, which had been tested in other inflammatory models, could have an effect on lupus phenotype and vascular dysfunction when administered as subcutaneous injections to these NCM2328 mice. And the results were, some of the results were more surprising than others. So I'll discuss them with you at this point. So these were treated chronically for a number of weeks, and then we assess autoantibody profiles, renal disease, and also vascular damage. So the surprising thing was that when we looked at anti-double-stranded DNA antibodies, in fact, the mice that had been exposed to chloramidine had higher levels of these antibodies compared to the vehicle mice that have not been exposed to chloramidine. But on the other hand, when we looked at complement activation and immune complex deposition, we saw that the mice that had been exposed to chloramidine had decreased complement activation and also decreased immune complex deposition, significantly decreased immune complex deposition. Here you're looking at IgG in the chloramidine-treated mice when compared to the non-treated mice, and here you're looking at complement deposition when compared to the non-treated mice. And this was associated also to significant decreases in renal inflammation. Now, how do we explain this difference between the autoantibody profiles and the immune complex deposition complement activation profiles? One hypothesis that, of course, we need to prove is that because this molecule may be interfering with immune complex formation as nets are not forming and complement activation is decreased, these autoantibodies are not depositing, are not involved in immune complex formation, and that's why the levels may rise. There may be other additional mechanisms, alternative mechanisms that we would need to explore, of course. The other findings that were the most striking in our study were those related to vascular phenotype in these lupus mice. So as I mentioned to you, lupus-prone mice, especially this model, but also the NCBW, have significant endothelial dysfunction, considered a surrogate marker of vascular damage. And we found that when we treated these mice with the chloramidine drug, there was a significant improvement in the capacity of the endothelium to relax in response to acetylcholine, suggesting that this drug was modulated vascular responses and indeed by preventing net damage and potentially by other mechanisms was vasculoprotective. And perhaps the most striking phenotype was related to thrombosis. And I mentioned to you that nets appear to be important in net formation, at least in venous thrombosis studies. We actually assessed arterial thrombosis doing an acute photochemical injury model where the mice are exposed to, are administered rose bengal, and then their carotids are exposed to a laser that induces a photochemical injury. And this leads to clot formation. And we typically measure the time to clot as an assessment of how prothrombotic these mice are. And we know that lupus-prone mice are prothrombotic, and specifically this model is prothrombotic and clots much faster than non-lupus-prone mice. So we took two approaches. And this was much shorter exposure. We only treated these mice for a week. So we either exposed them to vehicle, we exposed them to DNase, which would disrupt the nets, or we exposed them with chloramidine, which we think would prevent net formation. And what we found was very similar when looking at the DNase-treated mice and at the chloramidine-treated mice, in that the time to clot was significantly prolonged, and therefore these mice became much less prothrombotic. And when we looked at the clots in these mice, we found that if you look at vehicle, there were a number of cells, netting neutrophils, present in the clot, but the number of netting neutrophils was much decreased when these mice were exposed to chloramidine, implicating that this decrease in net formation may be one of the factors involved in the decrease in the prethrombotic phenotype by downregulating platelet activation and coagulation factor activation. And you can see here significant decreases in net formation in these clots. And the clots were much more bland, much less cellular than what you might find in the mice that were not exposed to the chloramidine. So, of course, this needs to be tested in other models, and more specific PAD4 inhibitors should be tested, and we need to further identify the exact mechanisms by which this compound is acting. We believe that significant mechanisms may be through inhibiting nets, but there may be alternative sources by which this drug may be active. So I want to finalize now by saying, and this is a very nice review that Felipe Andrade at Johns Hopkins recently published, where he explored the different models that may lead to loss of tolerance. And I don't think any of these models is mutually exclusive, and lupus being a heterogeneous syndrome, there may be different ways by which this is happening, but certainly aberrant cell death continues to be in all these models that he explores, where under some conditions apoptosis may be a key initial event, aberrant apoptosis. In other models, there may be a shared mechanism of impaired clearance to apoptotic cells or netting cells generated in lupus patients. It may be that in other patients or in other models, nets are a more central aspect of initial loss of tolerance and exposure of autoantigens, and the specific role that these low-density granulocytes, even present in the marrow, may play in initial stages with regards to exposure of autoantigens and tissue damage also requires to be further analyzed. So in summary, we propose that aberrant neutrophils may play prominent roles in lupus pathogenesis, at least in part through net formation, and that nets may serve as important sources of modified autoantigens that could trigger abnormal immune responses and organ damage in susceptible individuals. And it remains to be determined whether specific pharmacologic interventions that inhibit aberrant forms of net formation will be effective in autoimmune disorders. And again, the question is whether all nets are created the same way, whether there are specific proteins or specific post-translational modifications that may be particularly immunogenic in certain autoimmune diseases. And again, this goes beyond lupus. There are several reports now indicating that nets could be pathogenic in small vessel vasculitis. In Felty syndrome, we had a recent report in rheumatoid arthritis, and I think there is much more work to be done to explore the role of these cells and this process in autoimmunity. And I just want to end by thanking everybody involved, particularly current and former members of my lab that have done a lot of work trying to identify low-density granulocytes and the role of nets. My collaborators at the University of Michigan and in various departments and in other institutions. And thank you very much.
Thank you, Dr. Kaplan, for your excellent speech. And now we have a few minutes for questions.
Do you have a sense of the contribution of the net structure itself as opposed to the components in the nets? Since many net components are biologically active by themselves, such as histones and HMGB1, what does the net structure confer?
Yeah, I think that's a great question that has been difficult to study because many times when we want to explore the nets isolated from the neutrophils, we disrupt the structure of the nets. But I think this binding of DNA to various components present in the granules that may be protecting the DNA, for example, may be key. But I think we understand very little, and I think your point is excellent on how do you compare externalization of these molecules with degranulation or with other types of cell death. So I think it's something that needs to be investigated in more detail.
Thank you. I have two questions. Firstly, what do we know about the effects of drugs that we already use, corticosteroids, antimalarials, on net formation? And secondly, if a specific net formation inhibitor was available, what do you think the harmful effects of that would be on host immunity?
Yeah. So, but they're more excellent questions. I think we need to understand more how the drugs we currently use modulate net formation. I think there's preliminary areas that corticosteroids may not be potent modulators, but I don't think we have the final answer on that. And certainly antimalarials may be a very interesting target to study. We don't know enough about how these drugs are modulating net responses. We know very little about what modulates net responses. That's the problem. With regards to these inhibitors, I think, theoretically, like any, this is a process that is important in microbial trapping and potentially in microbial killing. So, but again, this goes to the question of whether all nets are created the same way, and if we could identify pathways that are particularly important in sterile stimuli leading to nets, that would be the way to go. What I can say is we did a very extensive characterization of what this compound was doing to other cell types, B cells, T cells, NK cells, monocytes. We did not see, at least in general, that the drug was affecting phenotype or function of these cells in the mice. And I think Mike Hollers has used this compound in collagen-induced arthritis and similarly did not find major effects on adaptive immune responses. But of course, this is early data and much more needs to be investigated.
Last question, please.
Yes. You mentioned that netosis is increased in kidney section and also in the skin of patients with lupus, as well as in the bone marrow of lupus-prone mice. Have you looked at if netosis is also increased in the secondary lymphoid organs, especially lymph nodes?
Yeah, no, we haven't looked. We are in the process of looking in mice. We are doing some studies now in human bone marrows from lupus patients, and we hope to have some data on that soon. But I think this is an excellent question, similar to what has been described about B-cell modulation by neutrophils in spleen, whether that's happening in lupus-prone mice.
Thank you, Dr. Kaplan, for these very interesting presentations. Thank you, Alfonso, and thank you all for coming.